What if I told you mastering the Indian National Biology Olympiad, the INBO, is just about asking the right questions? Welcome to the INBO Masterclass, where we dissect one question at a time so that you just don't memorize the concept, you truly learn them. INBO is the toughest exam of biology currently taking place in the country. The exam is not just about testing your knowledge. It is also about critical thinking. That is why this series is designed in such a way that it will develop a problem-solving mindset for the young minds of biology. In each video of this series, we will take one real INBO question, dissect it and analyze the concept behind it, and gradually you will develop a strategy where you can solve all similar types of questions. Most students struggle in the INBO not because of the lack of knowledge, but because of the poor strategy they apply. In each video, we will explore topics of cell biology, physiology, genetics, ecology, ethology, so that you will develop an Olympiad mindset for solving such questions. Each episode will be short, to the point and packed with insights. We will cover past year questions of INBO. Secondly, we will see some unique problem solving patterns, and finally some pro tips to solve questions in national and even international stages. This series is specifically designed for those students who have the basic concepts of biology quite clear and they are ready to level up so that they can prepare well for the international standard examinations. One such basic parameter I consider is the INBO paper analysis. This will give you a complete competitive mindset and you will be able to fight with the strong biology minds of the country. So if you are ready to think like a biologist, watch the first episode now and let's make INBO an easy journey. The first question is from the INBO 2024, that is last year's paper, and this is the question number 3. The question says, acquired immune system produces different type of antibodies upon exposure to an antigen. Now let's see which antibodies are actually produced by the acquired immune system. When an antigen exposure is done, mostly two antibodies are produced. One is the IgM and the other is the IgG. These are the two types of antibodies that are produced in case of any bacterial antigen or any viral antigen. The next part of the question says the type of antibody produced varies based on the time of initial exposure to antigen. That is the type of antibody produced. This will depend on different factor. First is the time of initial exposure, that is when the first antigenic exposure was done. Second is the duration after initial exposure, that is, say an antigen exposure took place 5 days back. So 5 is the 5 days post infection, we say DPI. This is the duration of exposure. And secondary or every subsequent exposure means, say a bacteria that entered my body today, it stayed inside my body for a few days. There was an ongoing infection. Say after 10 days, 20 days, the body was free of this pathogen. After 6 months, again that bacteria entered. This is called secondary or subsequent infection. A murine model, the next part says a murine model. Now what is a murine model? Although here in context of this question this is not much important, but since we are preparing for Olympiad, we need to know what a murine model actually is. Murine means for experiments we use laboratory animals like mice. So mice is considered as a murine model in biology. Was infected with bacterial pathogen, and as I said, in case of bacterial or viral pathogen we mostly see these two antibodies, that is the IgM and the IgG. These are the two antibodies that are usually produced. Serological analysis was carried out. Now, what is serology? Serology means the study of antigen antibody interaction. The other term we call it is immunology, but synonymously we can use we use the term serology instead of immunology. They are used alternately. In place of one another, we can use them. Serological analysis was carried out to monitor the presence of antigen specific IgM and IgG antibodies. As I said, since the infection was a bacterial infection, two antibody levels we will check in the titer process. One is the IgM level, the other is the IgG level. At different days post inoculation, that is, you can say this is the days post infection, DPI. We generally use this as a short form. Say a bacteria has entered today, 7 days from today we will abbreviate it as 7 DPI. DPI abbreviates as days post infection, as shown in the table. Now the IgM IgG levels are given in a table. We will see this table. So here it is denoted by some negative and positive signs, as you can see in this table, that the first sample is 1 day post infection, that is the first day. Say the bacteria has entered today, it is considered as the 0th day, and after that day is considered as the first day. Then the second one is given in the 7th days post inoculation. Then next one is given as the 10th day post inoculation, and the last one is almost 1 month, that is 28 days after infection or days post inoculation. So what is the actual relation between IgM and IgG? I will take a graph so that it makes the concept easier. So the graphical analysis is the best method to clarify about the IgM and the IgG levels in the body. So say this is a graph of the IgM that will denote the IgM and the IgG levels. First, let me check the IgM levels. For the IgM, say the graph somewhere goes like this. See the red one, the red line here is for the IgM titer levels in our body, and the second, say I will show it with green color, this is for the IgG in our body. Now a bacteria, say a bacteria has entered our body in this stage. If we examine this stage, here both IgM and IgG is negative. Clearly, when you go for test, you will get false results, because although the bacteria is present, the body has not yet formed antibodies. Because of this reason, if you go on the first day after infection, the level will show negative, the titer will show negative levels. As we can see here in this diagram, say this is the 1 day, the first day after inoculation, both antibodies are negative here. So gradually, if we see the changes, let's check the IgM. Here the IgM shows its peak. This is the this is the place where IgM shows its peak. The IgM value is very high, but if you see closely here, the IgG is absent. That is, here I can say this point is IgG negative. So although the infection is present, that is the positive IgM or the very high IgM levels and no IgG levels, it shows an ongoing infection. That is, the infection is present in the body. The next thing is, say if I take this point into consideration, say I take this level. So specifically, if I check this area, I will see that here the IgM level is gradually decreasing, whereas the IgG level is increasing. So I can say that this is the area where IgM is positive and IgG is also positive. The IgG is also positive. That is, this is also an indication of ongoing infection. That is, there are two cases where you will see ongoing infection. These are the two cases. The first case is where IgM is very high and IgG is absent. This is an indicative of the infection, ongoing infection. And the second case is where IgM and IgG both are positive, is a case of ongoing infection. These two are the cases of ongoing infection. Now, what does the term ongoing infection means? We can simply say that instead of ongoing, you can use this word active infection. That is, the bacteria is still present in the body. Now, how about I talk about this specific level? If you can see, how about I see this level only? I hope you can see this circle. So here, if I talk about specifically this point, see here the IgG levels, say let's write X here. In the region X, the IgG levels are very high, but IgM is absent. Now, what does that actually indicate? This indicates that although the IgG level is positive, very high, but the body is free of infection. Why this IgG level is high? This is because of memory cells. You know, our immune system has some memory cells produced by B and T cells. So this IgG level is high due to this memory cell condition. So here there is no active infection. We can clearly say that here there is no active infection. Since there is no active infection, that means this is a place. If I divide this graph into 4 areas, say these are the 4 areas. Say these are the 4 areas, first part, second part, third part and fourth part. If these are the 4 areas, we can clearly see that in the first part there is no active infection, in the second part there is ongoing infection, in the third part there is ongoing infection, and in the fourth part there is no ongoing infection. So ongoing infection is specifically on those areas, if I write them out, where IgM is very high or IgM and IgG both are positive. If the IgG levels are very high but IgM is absent, this is not an indicative of infection, and if both are absent, then this is also not an indication of infection. So now let's check what the question has asked. Which of the sample number in the given table talks about the ongoing active infection condition? Ongoing infection, the question is very clear now. Now it will be very easy. Now you just check the titer levels given here and answer. I will give you 5 seconds to think by yourself and then I will discuss the answer. It should be very easy if you have seen this lecture till now. So the answer to this question is ready. See the first case here, both IgG, IgM is negative. So this is clearly not a case of ongoing infection. So what are the questions where the ongoing infections will take place? If the IgM is very high, although IgG level is very low. This is one case, and the other case is where IgM is high and IgG is also high. So we can say that the second case, is a case of ongoing infection because IgM is very high and IgG is absent. And the third case is also a case of ongoing infection because both IgG and IgM levels are high. What about this last case? Here IgM is negative and IgG is double positive, means very high. Here we will say this is not a case of ongoing infection. Can you tell me why this is positive? If you have seen this lecture, definitely you can. This is because of the memory cell response. So the answer would be 2 and 3. Now where is the question? What are the options? So I hope this is clear from the question and it is done. 2 and 3. This would be the answer for these questions. So I hope you understand the concept behind the IgM, IgG fluctuating levels after a bacterial antigen has entered our body. I hope this video is very clear. I would continue this series of Olympiads with some short videos where I will analyze one question every day, dissect it as I said initially, so that you get a clear cut idea of the logic behind it and think like a biologist. If you are truly interested in learning the subject of biology with INBO preparation, this is the channel for you. If you really find this session helpful, I would ask you to stay tuned until we come up with the next video.